Mm. Okay, so hi, I am. This is the fourth week tutorial for the NPTEL course, I Think Biology, offered by the Azim Premji University. I am Geetika Agarwal. I'm a PhD scholar at the Center for Ecological Sciences in IIC Bangalore, and I'll be taking this tutorial today. Okay, so giving you a brief about what were the contents covered during this week. This week was all about cells. So they introduced what are cells. They talked a little bit about, they gave a few examples of different cells and discussed how they function. Then they talked about the cell cycle and how it's regulated and how cells move through the cell cycle and what are some checkpoints. And then in the end, there were a few lectures about the cancer biology and how cancer spreads through the body. Okay. So, coming to the first and foremost, the discovery of cells. So, how were cells discovered in the first place? So, there's this scientist called Robert Hooke who discovered cells in 1665. So, what he was doing is he basically had a microscope and he cut thin slices of bark or cork and he was observing them under a microscope. And he saw these tiny compartments, as you can see in this image, which gave a honeycomb appearance and they looked like tiny, tiny rooms. So he named them cells. And later he found out that these were actually the dead cells, and what he was seeing was the cell wall boundary of these dead cells in the cork. So, this is how cells were discovered. And moving on to the very basics of cells is the cell theory. And cell theory puts forth 3 points. One is that all living organisms are composed of cells. So, all of the living organisms, whether they be multicellular or unicellular, they're made up of cells. Okay. Next point is that cell is the basic unit of life. It's the basic structural and basic functional unit of life. So it's the smallest unit that forms living organisms. So it's the structural and functional unit of life. And all of these cells, they arise from pre-existing cells. So there are no cells that like just arise out of nothing. All of the cells, they arise from some form of a pre-existing cell or a living organism. Okay. So talking about the size of the cells. So how is the size of the cell, or what governs the size of a cell? Cells probably mostly range in the cells have sizes that are in the range of a few micrometers. So they don't grow very big. They are in the range of a few micrometers, and bigger cells are like the frog eggs or chicken eggs, which are a few hundred micrometers. So the cell sizes, they are mostly limited by surface area to volume ratio. So if you look at a cell, it's a 3D structure. So you can imagine it as a sphere. And you might already be aware that for a sphere, the volume will increase much faster than the surface area of the sphere if it has an outer membrane. So this surface area to volume ratio, it actually limits the surface area that's available for the exchange of materials in and out of the cell, and therefore this it makes it pretty difficult for the cell to do so if it has a uh, huge volume and a small surface area. For example, in this image, so the surface area of one large cube, it's given as 5400 micrometer square. But if in the same cube you fit multiple cells of like 10 micrometers in size, then the total surface area becomes very big of around 16,200 micrometers square. So that is why the size of the cell is actually limited by the surface area to volume ratio. Now, when it comes to cells, one can say that the shape of the cell can actually give you an idea about what function it plays or what is its role. For example, here's an RBC. An RBC is a red blood cell which is mostly flat and it does not possess a nucleus. And that helps it in performing its function of carrying oxygen and transporting oxygen throughout the body. Since nucleus takes up a big part of the cytoplasm or of the cell, so it does not have a nucleus, and also it does not have mitochondria, so that it does not have like any way of processing that oxygen that it carries throughout the body. So its cytoplasm is mostly composed of hemoglobin, and because it is flat, it can actually squeeze through very tiny capillaries, so it can easily perform its function of transport of oxygen throughout the body. Then there's an example of these pollen. Pollen, as you can see, come in various shapes, but most of them have these surface structures, which can help them actually attach to the surface of the stigma. So this makes them quite sticky. These ridges on the surface of it. So it makes it easier for the pollen to attach to the stigma, and that eases its way to reach the female gametes. And then there's this example of a nerve cell or a neuron, which is like an elongated cell, so that it can actually transport nervous signal or nerve signals or neuronal signals over longer distances through our body. So the shape of the cell can give you a little bit of an idea of what its function might be. Cells mostly have a very clear cut compartmentalization and a division of labor. So they have these different organelles which perform different functions. So, if we look at some basic organelles in a cell and what functions they perform, you can see that cell membrane basically acts like a gatekeeper, which looks at the or it allows certain molecules or certain signals to enter the cell and certain of them to leave the cell. But it keeps a check on what is entering and what is leaving through the cell. So it acts like a gatekeeper. Then there's the nucleus, which is the operation center. It contains the DNA, which is the instruction manual for the cell. So basically, the nucleus contains the DNA, and the DNA is mostly transcribed to make mRNA, and mRNA is translated to make proteins. So nucleus is where all of this is happening. It's kind of like the operation center. Then you come to mitochondria, which is actually the energy production unit of the cell. It's also called the powerhouse of the cell, because this is the organelle which is responsible for creating energy or producing energy for the functions of the cells. Then we have the endoplasmic reticulum, which can be thought of as a center where cargo is being produced. Cargo here mostly refers to the proteins that are produced in a cell. So it is the production unit where all of these proteins are being synthesized. Then there's the Golgi, which is the packaging center for this cargo or for the proteins. So Golgi sort of packages these proteins that are supposed to be transferred either out of the cells or to some other organelles within the cell. There is lysosome, which is present there for destroying the cargo or like the proteins that are not functioning properly or that are not you know uh, useful anymore, that are not required by the cell. They're actually destroyed by the lysosomes. So these are a few organelles. There's also the cytoplasm, which is basically uh, the milieu inside the cell, like the thing that makes up the inside of the cell between the nuclear membrane and the plasma membrane. That's the cytoplasm, and it is made up of a lot of things like the cytoskeleton, these filaments that actually help in the movement of the cargo via all of these tracks. So it helps in the movement of these proteins or these molecules within the cell. So cytoplasm is basically the inside of the cell, or the uh, the substance that fills up the cell between the nuclear membrane and the plasma membrane. Okay. Now, since we discussed about cargo and how it's being produced in the endoplasmic reticulum, packaged in the Golgi, destroyed in the lysosome, so I would like to talk about yeah. So I would like to talk about how protein is being trafficked in the cell, how it's being moved around in the cell. So basically, last week we discussed mRNA and how mRNA is actually translated to form proteins. So this mRNA is produced by the transcription of DNA inside the nucleus, and then it gets transported into the cytoplasm after being processed, after splicing and capping and tailing. After all of that processing, it's sent out into the cytoplasm. In the cytoplasm, it can either have two fates. It can either be translated by the cytosolic ribosomes, which are the free ribosomes present in the cytosol of the cell, in the cytoplasm of the cell, or it can get translated by the endoplasmic reticulum ribosomes. So, if it is supposed to get processed by the cytosolic ribosomes, which means that it will enter a non-vesiculated transportation. Basically, it will just be present in the cytoplasm. It can move around freely in the cytoplasm. And this cytosolic ribosomes, they process this mRNA, that they translate it into a protein. And this protein, either its fate is to become a cytosolic protein, which means that it stays within the cytosol in the cell and performs some function, or it gets transferred towards the mitochondria or nucleus. All of these places is within the cell, so it does not get transported out of the cell, or it's not like those glands which are secreting this protein out of them. So the protein that gets formed by the cytosolic ribosome, it has two fates. As I mentioned, it will either become a cytosolic protein and stay within the cytosol, or it can be max to max transported to the mitochondria or the nucleus. Other pathway that an mRNA can take is that it can get translated by the endoplasmic reticulum ribosomes. So what happens is, here the mRNA gets translated by the ribosomes that are attached to the endoplasmic reticulum. So you might already be aware from your high school that endoplasmic reticulum is of two types: either it's smooth endoplasmic reticulum or it's rough endoplasmic reticulum. The rough endoplasmic reticulum is named so because of its appearance, because all throughout its surface it has ribosomes attached to it. And the smooth endoplasmic reticulum does not have any such ribosomes attached to its surface. So these ribosomes that are attached to the surface of the endoplasmic reticulum, if they are translating the mRNA, then they form proteins that will be passed on to the Golgi apparatus, that will form a part of vesiculated transport system in the cell, or vesiculated protein trafficking will be happening for these proteins that are getting translated by the endoplasmic reticulum ribosomes. So these will then be packed into vesicles and transported to the Golgi apparatus, and from the Golgi apparatus they can either be sent to the plasma membrane, from where they exit the cell, they get secreted by the cell, they get secreted by the cell, sorry, or they become plasma membrane protein, which means the proteins that form a part of the plasma membrane, or they can either be sent to the back to the they can stay in the Golgi, or they can be sent to lysosome for destruction. So these are two fates that proteins come across when they're formed in the cell, and this is how this is what they can be subjected to. So, just to give a slight recap, an mRNA can either be translated by a cytosolic ribosome, which is a free ribosome floating in the cytosol, or by an endoplasmic reticulum ribosome, which means a ribosome which is attached to the membrane of the endoplasmic reticulum. If it is being translated by a cytosolic ribosome, it will either become a cytosolic protein, which is a free protein which stays in the cytosol, or it can be transported to the mitochondria or the nucleus of the cell. On the other hand, if the protein is being translated by ribosomes attached to the endoplasmic reticulum, then they enter the vesiculated protein trafficking. They get transferred to the Golgi apparatus, from where they can either be secreted out of the cell, or they can be transferred to the plasma membrane and become attached to the plasma membrane. They become a protein which is a part of the plasma membrane, or they can uh, or they can be transported back to the endoplasmic reticulum. They can stay within the Golgi, or they can be transported to the lysosome for their further processing in the cell. Okay, so these are some fates of proteins. Moving on to, I have been talking about vesicle mediated protein transport for all of these proteins that are processed by endoplasmic reticulum ribosomes. So, what is this vesicle mediated protein transport? So it happens between the endoplasmic reticulum, Golgi, plasma membrane, and lysosomes. So these are the four organelles that are involved in vesicle-mediated protein transport: endoplasmic reticulum, Golgi, plasma membrane, and lysosomes. And these vesicles in which these proteins get transferred, they have different coat proteins. So these coat proteins, they play an important role in the formation of the vesicles. These vesicles cannot be formed without the presence of these coat proteins. Okay, so one of these code proteins is COP1, and the COP1 is for retrograde transport. It is for the transport of proteins from the Golgi to the endoplasmic reticulum. So, COP1 is a protein which is required for retrograde transport of proteins from the Golgi to the endoplasmic reticulum. Then there is a COP2 protein, which is required for the opposite, which is the anterogate transport from the endoplasmic reticulum to the Golgi. And there is clathrin. Clathrin is a protein which is required for transport of vesicles from Golgi to the lysosome or from plasma membrane to the lysosomes. So basically, all the vesicles that are supposed to reach the lysosomes, they are coated by clathrin. So there are three types of these coat proteins: COP1, COP2, and clathrin. As I said, COP1 is for retrograde transfer from Golgi to the endoplasmic reticulum. COP2 is for anterogate transfer from endoplasmic reticulum to Golgi, and clathrin is for protein transport from Golgi to lysosomes or from the plasma membrane to the lysosomes. Okay. Now, moving on, this is an image which shows how vesicle mediated transport happens within the cell. So you have an endoplasmic reticulum here, which is marked by which is rough endoplasmic reticulum. It is marked by the presence of ribosomes in this red color. Then from here, as I mentioned, COP2, which is meant for anterogate transport from the endoplasmic reticulum towards the Golgi. So there are this vesicle has formed from the plasma membrane of the endoplasmic reticulum. It is coated by the COP2 protein. This is the vesicle here. It is coated by the COP2 protein, and then it gets transferred towards the Golgi here. So the COP2 protein covers this vesicle, and it pinches it off from the endoplasmic reticulum membrane, and this vesicle then gets transported to the Golgi. If the opposite is happening, if the transport is happening from the Golgi towards the endoplasmic reticulum, then COP1 is the protein which forms the coat. So COP1, it comes and attaches here as the coat protein, and it pinches this off from the Golgi. This vesicle gets transported and comes back to the ER. So this is the function of COP2 and COP1 respectively. Then, as you mentioned, there is clathrin. Clathrin is responsible to coat the vesicles that get transported either from the Golgi to the lysosome or from the plasma membrane to the lysosome. Okay, so one thing to keep in mind here, it's mentioned cis Golgi. So Golgi apparatus basically has two sides. The side that faces the ER, it's called the cis Golgi, and the side that faces the plasma membrane is called the trans Golgi. So Golgi has these two sides, cis Golgi and trans Golgi. Okay. So, moving on to what is the cytoskeleton and how it functions in the cell and why it is important. Okay. So, cytoskeleton, as the term suggests, it is the cell skeleton. So, like as humans, we have a skeleton of bones and muscles, or we have a skeleton of bones, and to it supports the muscles. That is what provides us our structure and our shape. So, as you might already get an idea from it, cytoskeleton is basically the skeleton of the cell. It provides the shape to the cell, it supports the cell, and it prevents it from being, from facing the pressure from the outside forces. It prevents it from getting destroyed by that pressure from outside sources, and it organizes the organelles inside the cell, and it helps in the transport of these molecules. It helps in cell division, and it helps in cell signaling. So these are the different functions of the cytoskeleton. And cytoskeleton is composed of these three elements: microtubules, intermediate filaments, and microfilaments. So, based on the size, microtubules are the largest, around 10 nanometers in size. Then, intermediate filaments are somewhere in between. And then we have the smallest one are the microfilaments. So, microtubules are made up of these small proteins which are called tubulin proteins. I will spell it out for you. They're made up of, sorry, they're made up of tubulin protein. And this tubulin protein has two forms. One of them is the alpha form, and one of them is the beta form. So all of both of these molecules of the tubulin protein, alpha beta, they alternatively join together, and then they form a tube of them, which makes these microtubules, which are actually uh, quite dynamic, and they keep forming and getting destroyed and reformed. Then there are these intermediate filaments, which form major part of the cytoskeleton, as you can see in this image. And then there are these microfilaments. So these are the components of the cytoskeleton. And the organelles, they actually get transported over these microtubules by the movement of proteins over these microtubules. So here is a small clip that shows you how organelles actually moves inside the cell. So you can see that here there is a kinesin protein which walks on a microtubule, as you can see in this video. So this is the kinesin protein, and it is carrying, this is the kinesin protein which is walking on the microtubule. It's a motor protein, and it's walking, I'm just using a term, but it is like getting progressed on that, and it is carrying a organelle along with it. So one side of the kinesin protein is attached to the cargo that it's carrying, and the other side moves forward on the uh, microtubule. Okay, so that is how organelles actually get moved inside the cell. So then the next lecture was where the professors discussed about their uh, favorite cells. So they gave a few examples of cells. So coming to the first one, so there is this cell called Toxoplasma gondii. Toxoplasma gondii is actually a single-celled parasite, and its definitive hosts are cats. Any form of cats, felines are its definitive host. What that means is Toxoplasma gondii actually enters felines. It can be ingested by felines, and there it enters into the intestines of these felines, where it actually forms gametocytes. It forms male female gametes. These fuse and they form a zygote, which matures into an oocyst, and this oocyst gets released with the feces of the cat. So this is the definitive host because sexual reproduction is happening in the cat and or in the felid host, and it does not really cause any infection. It does not actually cause any disease in the definitive host. And now this oocyst, which is released with the feces, it can actually be ingested by these intermediate hosts, which can be like birds or rodents, can even be humans at times. So when they are somehow ingested from the feces, these oocysts, they asexually reproduce in these intermediate hosts. So the cysts, they release this thing called bradyzoids. These bradyzoids, they're released in the stomach and the intestine, and they also release sporozoids that differentiate and they invade the tissue. And they invade the epithelial tissue and they start dividing asexually, and they cause some sort of a disease in these other intermediate hosts. And it's very interesting to know that the disease caused by Toxoplasma gondii is a type of a behavioral disease, and it actually affects the brain of the rodents, and it makes rodents um, less averse to felids. So it makes them approach these felids without any danger. They don't even get scared of, by the smell of the urine of a cat or things like that. And so basically, in a way, the parasite is providing more prey to its definitive host, which is the cat. So the cat is providing the parasite a place to sexually reproduce, and the parasite in return is actually making more prey available for the cat because it's affecting the prey behaviorally and making them less averse to these cats. It's making them more approachable. So this is a very interesting cell and a very interesting cycle through which the cell goes through and its life cycle. So it's interesting to see how such things exist in nature. Then the next thing that was discussed were diatoms. You can look at this image. They are these very pretty looking cells. They are actually microalgae, and they are photosynthetic. They can produce their own food by using sunlight. They can be present in fresh water, in marine water. They can be present in ditches, in wetlands, and they're actually very colorful, as you can see in these images. And why they're colorful is because in their cell wall they have silica, and this is actually called frustule. So this, they have silica present in the cell wall, which gives them such pretty patterns and colors, and this is called the frustule. These diatoms, they can either be free floating or they can be benthic, which means they're found at the floor of the river or the ocean, or they can be attached to other organisms. And they reproduce asexually by the process of fission, and their doubling time is approximately 24 hours. And why they are important is that they're actually the primary food source for a lot of fishes and invertebrates. Because as you can see, they're photosynthetic, they produce their own food. So they serve as a food for a lot of these um, uh, fishes and these invertebrates. Okay. So since they are autotrophs, they are photosynthetic, they provide food for these heterotrophs, which are fishes and invertebrates. They're responsible for about 20% of oxygen present on the planet because these diatoms are present in huge quantities in the ocean. So a lot of them process a lot of carbon dioxide and release a lot of oxygen. So not all oxygen comes from plants and from trees. A lot of it also comes from diatoms. They're very nice climate change indicators. And how they do so is that if there is a lot of increase in carbon dioxide, this carbon dioxide gets absorbed in the oceans and becomes carbonic acid. And this increases the acidity of the ocean. And with the increase in acidity, the amount of diatoms, the number of diatoms decreases. So they are very nice indicators of climate change. And they're used to make gaseous earth. Hi, am I audible? Okay. So I was actually talking about diatoms in this slide. So I was just mentioning how diatoms are important, and they are used to make diatomaceous earth, which is actually a very strong absorbent of water and is used in a lot of things like toothpaste and kitty litter and things like that. Okay. So the next cell that was discussed during the lectures of this week is actually Mixotricha paradoxa. Mixotricha paradoxa, as you can see in this image, it's actually a protozoan and it is found in the gut of termites. It has a symbiotic relationship with these termites. It gets protection and safety and a place to stay in the gut of the termites, and in return, it digests the wood for these termites. It's pear shaped and it's about 500 to 250 micrometers square in size. So M. paradoxa, very interestingly, has its own symbiotic bacterial species. So it has four of its own symbiotic bacteria. One of them lives inside the M. paradoxa and acts as a mitochondria. Now you would think that M. paradoxa, since it's a protozoan, it should have its own mitochondria. But in reality, M. paradoxa has actually done away with its own mitochondria. It does not possess a mitochondria on its own, and it actually uses this symbiotic bacteria inside it as the mitochondria. Another one is present, which is also present inside. It helps in breaking down cellulose and lignin, so it helps in digesting wood uh, by M. paradoxa. One of them acts as a flagellate. So spirochetes are bacteria like these small hair-like structures. These are spirochetes, which are bacteria present on the surface of M. paradoxa, and they can actually be controlled by the protozoan, and they help in the movement of the cell. And the last of them is forms these cilia, which help in controlling the directionality or in steering in which direction the cell is moving. So this is another very interesting cell which has a whole plethora of symbiotic symbiosis going around it. So it's symbiotic to the termite and it has its own four symbiotic bacteria in turn. So okay. So after that, in the next lecture, what was discussed was actually cell division and the cell cycle. And coming to it, first of all, I would like to talk about chromosomes and what really are chromosomes. So, DNA, which is present in the cell, as you can see here, this is like a short region of the DNA double helix, which is shown here. So, this DNA, it actually gets packaged in the cell. So, this DNA, these are like histone proteins, the ones shown here in pink. They are histone proteins, and around them, DNA gets wrapped around. So, it gives a bead on a string kind of a appearance. After this packaging, it gets further packaged into this helix. It forms 30 nanometer chromatin fiber, which is packed of nucleosomes. And then, so DNA wrapped around one histone is called a nucleosome. And when these nucleosomes are further packed, they form a 30 nanometer thick fiber. And then this gets packaged to form a 300 nanometer thick fiber, which is 10 times more condensed. And finally, it gets condensed into this mitotic looking chromosome, which you might already come across in your high school textbooks, that this is what a chromosome looks like or a chromatid looks like. And this here is the centromere of the chromatid. So, chromosomes are actually thread like structures which are made up of protein and a single molecule of DNA, and they carry the genetic information from cell to cell. So, humans have 23 pairs of chromosomes, one set coming from each parent. So we have 23 pairs of chromosomes. Half of them come from our mother and half of them come from our father. So again, this is how the DNA is getting packaged from the double helix, which wraps around these histone molecules, forms nucleosomes, and these nucleosomes get packaged further into chromatin fiber, which gets condensed into the chromosome. Okay. So let's talk about karyotype for a bit. Karyotype is actually just an image of an individual's complete set of chromosomes. So here is an image of a human karyotype. As I said, we have 23 pairs of chromosomes. So based arranging them based on their size and their homologous. So basically, this first chromosome here would be one that comes from mother and one that comes from the father. So one comes from each parent, as I said, in the pair. So we have these 23 total pairs from the largest to the smallest. And here these are the somatic chromosomes, and this is the sexual chromosomes, which is X and Y in humans. If it's XY, it's a male. If it's XX, both of them, then it's a female. Okay. So this is the karyotype of a human male. So now coming to the cell cycle, having talked about what is actually a chromosome, we'll talk about the cell cycle. So, cell cycle is actually the sequence of events between the formation of the cell and its division into daughter cells. So, when a cell gets formed and then grows and its DNA gets synthesized and then it gets divided further into daughter cells. So, that is the entire cell cycle. So, in this part of the lecture, we'll discuss why do cells divide, what triggers this division of cells, how is cell division studied, and the regulation of the cell cycle. Okay. So first of all, why do cells divide? So why do you think cells undergo cell division? Okay. So to give a few reasons, cells undergo division for four main purposes. That's for reproduction, which can happen, say, in the case of binary fission, where a lot of single cellular bacteria and things like that, they just divide to reproduce. It is for growth and repair of tissue. For example, in humans or in general, when an organism is increasing in size, the cells are not really just growing in length, they're actually dividing to form multiple cells, and that is how we increase in size. For repair, for example, say you scratch your skin, after a few days you see that new skin cells have actually repaired that scratch. So that is happening by via the division of the skin cells. And then we require them for gamete formation, which is actually required for reproduction in multicellular organisms, where you require gametes. For example, sperms and ova uh, in animals. So there are three different types of cell divisions that we'll be discussing and that actually exist. One of them is called binary fission, mitosis, meiosis. You might already be aware of this because all of this is a lot of the times it's covered in the high school. But to give you a slight overcap, so what is happening in binary fission is that it occurs in single celled organisms, for example, bacteria, and it creates identical cells and it begins with DNA replication. The number of chromosomes stays the same, and binary fission serves the purpose of reproduction of a bacteria and it creates entirely new living organisms. Mitosis is very similar to binary fission, but it occurs in multicellular organisms. This also creates identical cells which have the same amount of DNA, but it serves the purpose of growth and healing in multicellular organisms. It occurs in somatic cells and includes these different steps called prophase, metaphase, anaphase, telophase. Then there is meiosis, which is slightly different from these two. Again, it does occur in multicellular organisms, but it creates haploid cells, which are called gametes or sex cells, which are then fused again to form the zygote. Again, it begins with the DNA replication, but the number of chromosomes is reduced to half. So the number of chromosomes is just half in the resultant daughter cells. Meiosis, as we mentioned, because the chromosome number is half, it produces haploid cells, which are cells with only half the amount of original DNA from the parent cell. Serves the purpose of reproduction in multicellular organisms, so it is required for the purpose of reproduction. And it includes the following steps. So, there are some similarities in all of these processes. All of them produce new cells, all of them lead to a replication of DNA, and all of them play a very important role in the life cycle of the cell. Okay. So this was a slight overview of how what are the different types of cell divisions, And so, two things to keep in mind is that binary fission mostly happens in single celled organisms, leads to the production of clones or same cells with the same DNA, and they serve the purpose of reproduction in single cellular organisms. Mitosis happens in multicellular organisms. It also leads to production of identical cells with same DNA, but it serves the purpose of growth and healing in multicellular organisms. Meiosis serves the purpose of reproduction in multicellular organisms, and the resultant cells are actually haploids. They have only half the amount of original DNA. Okay, so moving on to what binary fission looks like. So this is how, uh, say, a bacterial cell looks like. These are ribosomes here. This is a packaged DNA. And as we know, prokaryotes do not really have a nucleus or a nuclear membrane. So there's just a DNA bundle here, and this is a plasmid. So what happens is the DNA gets duplicated. So there are two DNAs. The plasmid is getting duplicated. And then each plasmid attaches itself to a DNA, or gets allotted to one of the DNAs, and then the cell divides. There's a ridge formation, and then cytokinesis really happens, and then one cell has one DNA, other DNA. Even though it's shown in different colors, both of these are exact copies of each other. So this is the very basic of binary fission. Then you move on to mitosis. In mitosis, these are actually chromosomes that we discussed. The DNA has gotten packaged entirely, and these chromosomes, they get duplicated. So there is DNA replication happening in the beginning. They get duplicated, then they arrange themselves on these spindle fibers, and then they get separated, and two diploid cells. So these cells, as you can see, the daughter cells, are identical to the parent cell entirely. So this is how mitosis happens, and there are different phases in mitosis that we will discuss in detail. But first, let's talk about the cell cycle. So in cell cycle, there are these different phases. There is this M phase, where mitosis is, or the cell division is actually happening. Then the cell can either get into the G naught phase, which is the resting phase, where it is out of the cell cycle, or it can enter the G1 phase, where the cell grows. And then there's this first checkpoint. Then there is this S phase, where the DNA gets made or replicated. And then there is the G2 phase. And then after this, the cell again enters the cell division phase. There are these multiple checkpoints in this cell cycle, which we'll be talking about in the next slide of cell cycle regulation. So these phases, as I said, there is a G naught phase, which is the resting phase, where the cell is actually outside this cycle. There is the interphase, which is the preparatory phase. So all of this G1, S, and G2, they together form the interface. So from here to here, this all is actually the interface. And then where the cell is actually preparing for cell division. And then there is the M phase, which is the mitosis phase or the cell division phase. Okay. So moving on. Okay, yeah. So as I said, in G1, the cell actually grows, and it checks if there is any sort of a damage in the DNA or something like that. And then after it's crossed the first checkpoint, in the S phase, the DNA starts duplicating. And in the G2 phase, all the other organelles also start duplicating, so that the cell can now divide. Okay. Because you need a copy of every organelle and the DNA for the cell to actually divide and form two daughter cells, which are exactly identical to each other. So, coming to mitosis in a little bit of more detail. So, there are these different phases that we talked about. One of them is the interphase, and these are the different phases in the M phase, which is the mitosis in reality. So, interphase is the time when the cell is actually preparing to undergo cell division. It increases in size, and it replicates its DNA and its organelles, and it prepares for cell division. So they have marked these different parts inside a cell. There is this nucleolus, there is the nuclear membrane, there is these duplicated chromosomes. So you can see the chromosomes, yellow and blue, have been duplicated. There are two copies of each. There is the nucleus, and then there is the centriole. So what happens in the prophase is that the chromosomes, they get condensed. So here you can see they are like fiber like, and here they are actually condensed into these structures. So the chromosomes, or the genetic material, they condense, they coil tightly, and the nucleolus disappears in the prophase. After prophase and before metaphase comes a phase called prometaphase, where actually the nuclear membrane also disappears, and these centrioles move to the opposite ends of the cell. Then in the metaphase, these centrioles, they produce this spindle fiber, as is shown in the dashed lines, and the chromosomes actually align in the central plane of the cell. Like here, you can see them aligned. So they align on this equatorial plate, so they attach to the spindle fibers via their kinetochores or their centromeres, and they align on this central equatorial plate. Then comes the anaphase. You can remember A from apart, where these chromosomes actually get pulled apart by the two poles of the centrioles. So these centrioles, what they start doing is that they start pulling the spindle fiber, and as they're pulling the spindle fiber, the chromatids also get pulled along with them. So the chromosomes also get pulled apart to the opposite ends of the cell. So you can see the cell gets an elongated appearance because the centrioles move further apart from each other and they're pulling these spindle fibers along with them. So the chromatids separate and they move to the opposite ends of the cell. Then comes the telophase. In the telophase, the two new nuclei get formed, and they form around each set of the chromosomes, and the chromosomes, they decondense, and these cells, they start to split. So you can see this ridge here, the cell has started to split, and then happens the real cytokinesis, where the cytoplasm gets split into two different daughter cells. So the cytoplasm gets split into two identical daughter cells. So these are the different phases of mitosis. Interphase is the preparatory phase. Then is the prophase, where the chromosomes, they condense, and the genetic material, it basically condense, coils tightly, the nucleolus disappears, the nuclear membrane disappears, centrioles move to the opposite ends. So this centriole, it actually divides into two, and they move into the opposite ends. Centrioles move into the opposite ends, they make spindle fibers, and during the metaphase, the chromosomes attach to the spindle fibers and they arrange themselves on the equatorial plate. In the anaphase, they get pulled apart to the opposite sides of the cell. And telophase, the nuclear membrane forms again, and the cell starts to divide. And then cytokinesis, the cells actually really divide. So here's a short video about mitosis where you can get an idea of how this is all is happening.
During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell.
So this video explained how mitosis really happens and what happens during the different stages. So as mentioned, during the prophase, basically the chromosomes, they condense, the nuclear membrane, the nucleolus disappears, and spindle fibers start forming from these two structures called centrioles. During metaphase, these spindle fibers, they attach to the kinetochores, which are present on the centromeres of the chromatids, and these chromatids arrange on the equatorial plate. In the next phase, these centrioles move further apart, and they're also pulling the chromatids, sister chromatids, apart by shortening of these spindle fibers. And then in the telophase, the nuclear membrane gets formed again, and then cytokinesis really happens. So these are the different steps of mitosis. So moving on to the next form of cell division, which is actually meiosis, in which, as we discussed, a diploid cell, which means it has 46 copies of, so as you said, 23 pairs, right? 23 pairs of chromosomes. One set comes from the mother, one comes from the father. So diploid cell has 46 of these. So in meiosis 1, this gets halved, and then all of them form haploid cells. So basically all of them in the end, they have only 23 copies, not 46, just half the number of it. So what is happening here is again an interface. They're getting prepared and they double the amount of their DNA replication, which happens. Then homologous sister chromatids, the homologous chromatids, they come along each other, and then they exchange some during meiosis one. They exchange some of their genetic material within each other. So you can see that the gray ones have exchanged genetic material with the red ones, and so on has happened here also. And then it divides again to form daughter cells. So I will explain this in more detail. So I've been mentioning homologous chromosomes. So homologous, basically, if I go back to the karyotype, from here you can make, you can see, as I mentioned. So these are actually homologous chromosomes in one where there is a pair. So the same pair, the same chromosome, say number one, which comes from the mother, and number one, which comes from the father, they are actually the homologous pair. Number two that comes from mother, number two that comes from father, this is the homologous pair of chromosomes. So these are the ones that actually come together and they exchange some genetic material between each other during meiosis. Okay. So for meiosis also, I would like to show you a short video which will explain uh, how meiosis really happens in a very nice way. So yeah.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Let's look at what happens during meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with four genetically different haploid daughter cells, each containing.
Yeah, so this was a video which explained how meiosis happens, and so I hope this is clear to everyone because it's very important to know how meiosis is actually happening. So what we saw is, um, yeah, so basically it results in haploid daughter cells which are actually genetically different from each other. So in prophase 1, when actually the sister chromatids are going through, or the homologous chromosomes are going through exchange of genetic material. Prophase 1 is further divided into four different, or actually five different stages. So prophase 1 actually has this first stage called leptotene. In leptotene, what happens is that the chromosomes, they condense. So you can see these homologous chromosomes, one in pink and one in blue. So one basically comes from the mother, one comes from the father. So the chromosomes condense. Then the chromosome pairing happens. So in zygotene, the chromosome pair, since these are homologous chromosomes, they come together and they form something called the synaptonemal complex where they're actually attached to each other. So the pairing happens and synapsis begins. In pachytene, the chromosomes, they get fully synapsed. Like basically they join on this one end, like they're sort of connected on this one end, and the crossing over begins. So they exchange genetic material. As you can see in this area, they're exchanging genetic material. So that happens in pachytene. So in leptotene, chromosomes condense. In zygotene, the synapses actually begins and the pairing happens of the homologous chromosomes. And pachytene, the crossing over actually really happens. And in diplotene, the synaptonemal complex, it starts to disappear. So you can see this light pink color, it has started to disappear. And the chiasmata or the chiasma is now visible. Chiasma is actually the site where crossing over really occurred. So the chiasma is now visible. And then there is diakinesis. Diakinesis is basically when the crossing over has completed, and you can see further the chromosomes have contracted even further and formed these two homologous chromosomes. Okay, so these are some stages in the prophase 1. So coming to how the cell cycle is getting regulated, how the cell knows when to do what. So cell division is not really just happening over and over again without any control over it. There is actually a correct time and a correct duration after which the cell undergoes division. It cannot just undergo division just after getting out of mitosis or cell division. So here you can see that they have marked three different checkpoints. One of them is called the cell growth checkpoint, which is the first checkpoint. Then there is the DNA synthesis checkpoint during the S phase, and then there is the mitosis checkpoint, which happens before the cell enters into the M phase. So what is happening here is, number first is the cell growth checkpoint, which happens after the G1 phase. Here, what is happening is, as I mentioned, it happens at the end of the G1 or the growth phase. And here, the cell checks if the cell is big enough now, and all of the proteins have been made which are required for the synthesis phase. So, in the synthesis phase, as we know, the DNA is supposed to duplicate, and all of this matter is supposed to form. So, you require the necessary proteins for the DNA duplication to happen. So, in G1 phase, it is checked that the cell is now big enough, it has grown, and all of these proper proteins are available. If any of these is the issue, then the cell, or if the DNA is not proper, then the cell, it enters the resting phase until it is ready to divide again or ready to enter the cell cycle. But if they see that the DNA actually has is problematic, there is some issue in the DNA, it has not been replicated properly, then it might actually undergo apoptosis and the cell might just die if the division in the previous cycle did not happen properly and the DNA is not proper. But otherwise, it can enter just into the resting phase where it keeps growing. So this is the number first cell growth checkpoint, where it is checked if the cell is big enough and if all the major proteins are present required for the S phase. Then during the synthesis phase, there is this DNA synthesis checkpoint, and as the term suggests, it checks if the DNA has been replicated properly and correctly. If this happens, then the cell continues into the mitosis phase or continues on into the cycle, and then comes the mitosis checkpoint. This checkpoint, it occurs during the mitosis phase, or I would say a little during the early half of the mitosis phase. It checks if the cell is okay to enter the um, if all the it happens actually before cytokinesis, and it checks if the mitosis has completed properly and if all the chromosomes have separated properly, and if so, then the cell can actually divide, cytokinesis can take place, and the cycle repeats itself. So these are the three checkpoints that are present that regulate cell cycle. Now you might be wondering how actually these checkpoints work, how like what is really going on in the cell, how they're actually checking these things, that if what are the molecular players in the cell cycle regulation. So coming to these checkpoints, the molecular players here are actually the cyclins and the cyclin dependent kinases. They play a very important role in the cell in passing a checkpoint. Cyclins are actually proteins, and cyclin dependent kinases are also proteins, and kinases are responsible for phosphorylation of other target proteins. So kinases are actually enzymes, and these enzymes, they phosphorylate specific target proteins. And for their activation, they are dependent on the proteins called cyclins. That is why they name cyclin dependent kinases. So these two players, cyclins and cyclin dependent kinases, are important for a cell crossing its checkpoints. So in this image, you can see if there is no cyclin present, then these cyclin dependent kinase is actually inactive, and none of the target proteins, they get phosphorylated, and so all these factors are actually off. If you now add the specific cyclin, in this case, since we're talking about S phase, so if you add the G1 or the S cyclin, it will come and attach to the CDK. It will activate the cyclin dependent kinase, which will lead to phosphorylation of the target proteins, and then the cell can cross the first checkpoint, and the S phase can begin, and the DNA replication can actually happen. And this is what the phosphate groups look like. So this is how the checkpoints are crossed. So, what are the different cyclins that are required? So, you can see here, this is how the concentration of the cyclins vary throughout the cell cycle. So, as the growth phase starts, the G1 cyclin starts increasing, and it only decreases when the cell has actually entered into the M phase. Till then, it stays more or less constant. Then, talking about the first G1 or slash S cyclin, this is the cyclin which helps the cell cross over from the G1 phase to the S phase. So, this is for the first checkpoint, the G1 checkpoint. So, this is the G1S cyclin. So its amount starts increasing when the cell is supposed to cross the first checkpoint, and as soon as the cell has crossed the first checkpoint and entered the S phase, then the G1S cyclin gets destroyed. Then you come to the S cyclin, which helps pass the second checkpoint, which checks whether the DNA has been replicated properly. So it starts increasing a little before the S phase and during the S phase. And when the checkpoint has been crossed, that okay, the DNA has been synthesized properly, then S cyclin also starts to go down in amount. Then there is this last one, the M cyclin, which is for the last checkpoint, which is the mitosis checkpoint. Again, its amount starts increasing and it's destroyed after it is not required. So these cyclins, as the term suggests, they're also produced in a cyclical manner during the cell cycle. Okay. Talking about inhibitory phosphorylation. So the CDKs, as we mentioned, they're activated by cyclins. If the cyclins are present, they're activated. But there is also something called inhibitory phosphorylation. So the CDKs, they have certain sites. So if they're activated, for them to be activated, they need to be phosphorylated on one of these particular sites. But they can also be negatively regulated, and they might get and this might happen by phosphorylation of these other sites. So as you can see in this image, so this is a cyclin CDK, and it is inactive because it is not phosphorylated at the correct sites. So there is this protein called CAK, which is going to activate it. Since you can see in this arrow is for activation. And there is this another protein V1, which actually phosphorylates it on the other side, which makes it inactive. So there are these two sites here. When the CDK has been phosphorylated on both the sites, it is actually inactive, and this will prevent the cell from crossing over that checkpoint. But in this case here, CDC25 is another protein which actually um, dephosphorylates this particular protein, and it converts it back into an active protein which has the phosphorylate or the phosphate group only on one side, which is the active site. So CDC again leads to activation of the protein. So V1 is phosphorylating CDK and making it inactive, and CDC here is dephosphorylating that particular site, which makes it inactive and making it active again. So it might be a little confusing, but this is how it happens in the cell. So yeah. So V1, just remember that V1 actually inactivates the CDK and CDC25 actually activates the CDK. Okay. Now, coming to the importance of cell cycle, now that we've discussed the entire cell cycle, the regulation of the cell cycle, mitosis, meiosis, why is the cell cycle important? It can help you determine the size of the cell or the cell size, in which, depending on which stage of the cell cycle you are at. It can help you understand cell division and how cells grow and develop. And if the cell cycle is actually impaired, it may lead to a lot of diseases. For example, if the cell cycle is not happening properly, if the checkpoints are not crossed properly, it might lead to cancer, where the cell just keeps dividing uncontrollably. So, yes, this brings us to cancer. This was the last topic that was discovered during this week. So, what happens in cancer, as you might already be aware? So, there is a loss of control over how cells are dividing, over how proliferation of cells is happening in the tissues and organs of the body. So cells, they grow uncontrollably and they start spreading to different parts of the body. So as I'm talking about they're spreading to different parts of the body, it means that there is also a loss of control over the restriction of movement. So in general, when cells are in an organ, they are attached to a basement membrane, and there is a way, and they are also their division is also inhibited by a process called contact inhibition, which we'll come to later. So contact inhibition is actually where cells, when they're in contact with some other cell, so if the cell membrane of a cell is in contact with all the neighboring cells, it restricts it from dividing further, because the cell knows that okay, we're already crowded here, so it does not divide. That's called contact inhibition. But cancer cells lose this form of inhibition of cell division, and they keep on dividing uncontrollably. And there is also a loss of control over the restriction of movement. So the cells can also move away from the organ in which they're meant to be. As I mentioned, there is loss of adhesion from the basement membrane, because that is what keeps the cell in place. And there is loss of stromal and parenchymal compartmentalization. I will mention a little in the next slide what is actually stromal and parenchymal compartmentalization. So you can understand the pathology of an organ in terms of how it's getting, the cell arrangement in the organ is getting disrupted. So you can see that the organ is diseased because you will see that the arrangement of cells in that organ is actually gotten disrupted. So cancer can be also seen as a problem of pattern deformation, how the cell pattern has been deformed from what it was supposed to be. So, what are the components that help in maintaining structure? So, as we talked about cytoskeleton, which is the skeleton of the cell, it actually provides a scaffold that shapes the cell and it allows it to interact with its milieu, which means its surroundings. And cells can be connected together via different junctions like cell to cell junctions, cell matrix, gap junctions. These are some different types of junctions by which neighboring cells are actually connected to each other. Gap junctions allow for the exchange of materials and signals between these neighboring cells. Adherence junctions, as the term suggests, they'll allow the cells to be tightly bound to each other. Hemidesmosomes are actually junctions that allow the cell to be bound to the cell membrane or the basement membrane of the organ. So here you can see, so these are cells in blue here. This is the basement membrane in turquoise. And then these are the different junctions. So as you can see here, here is the tight junction. So you can see how the membranes are actually attached to each other. So tight junctions are actually happen by the interlocking of junctional proteins. So this plasma membrane of both the cells, they have junctional proteins. And so we were talking about the different types of cell junctions that are present. So I was mentioning there are these tight junctions. So these plasma membranes of adjacent cells, they actually have these junctional proteins which get interlocked together, leading to these tight junctions, which keeps the cells bound together. Then there are these anchoring proteins or these anchoring junctions, which have these protein filaments that actually again lead to the cells being tightly bound together. There is these hemidesmosomes, which I mentioned, which link the protein, which link the cell to the basement membrane. And there are these desmosomes, which actually link the two cells together with each other. And there are these gap junctions, which have channels between the cells, which lead to transfer of material and exchange of materials and signals between the two cells. Okay, so these are the different types of junctions that are present in the cell. Moving on to extracellular matrix, what really is the extracellular matrix? So it is actually the collection of proteins which are secreted by cells, and it's the um, and it houses the cells and it packs them and provides them support and cues. The extracellular matrix is actually secreted by other cells also that are the part of the connective tissue and they travel between tissues. So ECM is actually has two parts. It has one part which is secreted by the cells within the organ or the native cells, and it has the other part which is secreted by the cells of the connective tissue or the cells that travel between these tissues. So this organ parenchyma is actually the part of the extracellular matrix which is secreted by the native cells or the organ cells of the organ, and the stromal part is the one that is secreted by these connective tissue cells. So this is what a normal extracellular matrix looks like. So you can see here there are different parts. There is the epithelial cells. There are these different types of cross-linked collagen proteins. There is these proteoglycans, and then there is these endothelial cell layer and the basement membrane here in blue. But you can see that in cancer, everything gets very stiff and hi, sorry for the disruption. I seem to have lost my internet connection again. Sorry for the patchy internet. Okay, so I will leave my video off for some time now so that the internet does not get disrupted. So we were talking about how the what is the extracellular matrix. And then let's talk about metastasis, which you might already be aware is a very important concept. It's a very important term when it comes to cancer biology. It's actually metastasis refers to the movement of cells from one organ to other, and it leads to the morbidity of cancer. So when cells actually move out of the organ where they belong to and enter some other organ, it actually makes it even worse, and they cause tumor in new areas. So the spread of cancer, it's not completely chaotic, but this also involves some underlying principles of movement. And by studying metastasis, you can actually understand how these cancer cells are traveling and moving across the body. There are these different steps in metastasis. The cancer cells, they move out of the local organ, they seek out blood vessels, and they enter the blood vessel, and they travel via blood, and eventually they exit the blood vessel and they enter into a new organ, and they start spreading in that new organ. So a lot of the cancer cells, they actually die while traveling through the blood, but some of them, which do survive, they can exit the blood vessels at different points, and they can enter into the new organ and start spreading in this organ. So here's a short video to show you how metastasis really occurs.
Most cancer deaths are caused by metastatic cancer. Metastasis occurs when cancer cells break off from the original tumor, which is also called the primary tumor, travel through the body, and begin to grow in other tissues and organs. Metastasis causes death by damaging important organs, such as the brain, lungs, and liver. Cancer cells continually break off from the primary tumor. Some of these cells may move into nearby blood vessels and lymph vessels. Cancer cells can travel along these vessel highways to other parts of the body. Many cancer cells die while traveling through the blood and lymph vessels, but some may survive and stick to the vessel wall, then move through the vessel wall into another body tissue. These cells may then divide and form a metastatic tumor. Where are cancer cells most likely to spread? Cancer cells tend to spread to certain places in the body depending on the site where the tumor first formed. For example, breast cancer tends to spread to the bones, lungs, liver, and brain, and colon cancer tends to spread to the lungs and liver. It's important to remember that a metastatic tumor is the same type of cancer as the primary tumor. In this example, colon cancer cells have traveled through blood vessels and formed metastatic tumors in the liver. These metastatic tumors are colon cancer, not liver cancer. Metastatic colon cancer is usually treated with drugs and other therapies used to treat colon cancer. Researchers at the National Cancer Institute and other research organizations are studying metastasis and ways to prevent it. Preventing metastasis will help prevent many cancer deaths.
So this was all from this week. In this week, we mostly discussed, we gave an introduction to cells, talked about different types of cells, cell cycle, how the cell cycle gets regulated, what are the different, how protein gets trafficked through the cell, all of these concepts, and then we discussed a little bit about cancer biology. Coming to the question answer session for this week. So the first question in the quiz is that which organelle is known as the powerhouse of the cell? So this is a very simple question. Which organelle is known as the powerhouse of the cell? Nucleus, mitochondrion, ribosome, or the Golgi apparatus? I will give the answer after 5 seconds. Yeah, I will put a timer for 10 seconds, and then, yeah. Which organelle is known as the powerhouse of the cell? The correct answer to this is actually the mitochondrion. Mitochondrion, as you might already be aware, and as I mentioned during this lecture, is called the powerhouse of the cell. It is responsible for the formation it's responsible for generating energy in the cell by breaking down ATP. Okay, what is the main function of the nucleus? What function does the nucleus play? Is it for protein synthesis? Is it for controlling cell activities and storing the DNA? Is it for producing ATP or for transporting molecules? Again, I will give you 10 seconds and then I will tell you the answer. Yeah. So the correct answer to this is that nucleus is required for controlling cell activities and for storing DNA. Question number 3: which structure is found in plant cells but not in animal cells? So, which of these structures is actually found in the plant cell and not in the animal cell? Is it the mitochondria, or the cell wall, or the ribosomes, or the endoplasmic reticulum? Which structure is found in the plant cells but not in the animal cells? So, the correct answer is cell wall. What are the two main phases of the cell cycle? So, what two main phases of the cell cycle? Is it interphase and mitotic phase, G1 and G2, prophase and metaphase, or DNA replication and cell division? Which of these are the two main phases of the cell cycle, or in which the cell cycle can be divided? Yeah. So the correct answer to this question is actually the interphase and the mitotic phase, as we already discussed. There are two phases: interphase, mitotic phase. Interphase has the G1, S, and G2 phase, and the mitotic has further prophase, metaphase, anaphase, telophase. Yeah. So, during which phase of the cell cycle does DNA application occur? So, during which of the phase of the cell cycle does DNA replication really occur? Is it the G1 phase, the S phase, the G2 phase, or the M phase? So, which of these phases uh, is during which the cell cycle the DNA replication occurs? Yeah, so the correct answer to this is actually the S phase, which is also called the synthesis phase, that is during which the DNA replication occurs. What happens in the G1 phase of the cell cycle? So, what really happens during the G1 phase of the cell cycle? Is it DNA replication, or does the cell grow and prepare for DNA synthesis? Do the chromosomes align in the center of the cell, or does the nuclear membrane disappear? So, which of these followings happen during the G1 phase of the cell cycle? Yeah. So the correct answer to this question, as you might already be aware, is that the cell grows and it prepares for DNA synthesis, as we already discussed during G1. It is during the S phase that the DNA replication really occurs, and chromosomes align in the mitosis. This is during the metaphase, and the membrane disappears during prometaphase. What is the function of checkpoints in the cell cycle? So, what is the function of these different cell cycle checkpoints? Is it to allow the cell to skip phases? Does it allow the cell to skip the phases? Does it ensure the proper progression through the cell cycle? Does it stop mitosis, or does it duplicate organelles? So, what do you think is the function of checkpoints in the cell cycle? Yeah, so the correct answer is that it ensures the proper progression through the cell cycle. So it ensures that the cell properly progresses through the cell cycle. So what happens if a cell fails a checkpoint and cannot repair the damage? Does the cell continue to divide normally? Does it undergo apoptosis, or the programmed cell death? Does the cell skip the next cycle or the next phase, or does it increase its size? So what happens if a cell fails to cross a checkpoint and cannot repair the damage? So the correct answer to this question is that the cell actually undergoes apoptosis, or the programmed cell death, if it cannot really repair the damage. What is the purpose of mitosis? What purpose does mitosis serve? Does it produce gametes? Does it lead to genetic diversity? Does it divide a single cell into two identical daughter cells, or is it just to replicate the DNA? So, what is the purpose of mitosis? So, yeah, the correct answer to this question is that it leads to the division of a single cell into two identical daughter cells. So, that is what a mitosis does. In which stage of mitosis do chromosomes align at the center of the cell? Prophase, metaphase, anaphase, telophase. So, what is the phase at which cell cycles actually align? Sorry, chromosomes actually align at the center of the cell. Prophase, metaphase, anaphase, or telophase. So the correct answer to this question is the metaphase. It's at metaphase that the chromosomes align at the center of the cell. They are attached to the spindle fibers via the kinetochores, and then they align on the equatorial plate. And in the next, which is the anaphase, they get pulled apart. Which of the following correctly matches the mitotic phase with its events? So, which of these following questions is it correctly matched? So, in the prophase, do chromosomes decondense? In the metaphase, do chromosomes separate? In the anaphase, are the sister chromatids pulled apart? In the telophase, does the nuclear membrane start to dissolve? So, you've got 10 seconds to think about this answer. So, what happens? Which of these is correctly matched to what really happens in the event? Yeah. So the correct answer is that it's in the sorry. The correct answer is that the in the anaphase the sister chromatids are actually pulled apart. Yeah. Moving on to the next question. What is cancer? So what really is cancer? Is it the normal part of a cell cycle? Is it the uncontrolled division of cells? Is it a type of virus or is it a result of mitosis? So what really is cancer? Yeah. So the cancer is actually, as we discussed, uncontrolled division of abnormal cells. You might also already be aware. Which of the following is a characteristic of which of the following is a characteristic of cancer cells? Is it controlled cell division, sensitivity to cell cycle checkpoints, loss of contact inhibition, or a shorter lifespan than normal cells? So what happens during what is the characteristic of cancer cells? So the correct answer is loss of contact inhibition. They are no longer able to stop dividing after coming in contact to neighboring cells. They keep on dividing. Cells cannot grow very big. Why? Do they find it difficult to find material? Do they metabolically not remain active? Is it the surface area volume ratio is a limiting factor? Or do they evolve to be, have they evolved to be small? So, what is the reason that cells cannot grow very big in size? So, as discussed during this lecture, it's actually the surface area to volume ratio, which is a limiting factor because if a cell keeps increasing in size, its volume will keep expanding, but the surface area will not increase quite as much to properly let the exchange of materials happen. What are some challenges in studying cancer and finding appropriate therapies? So, what are some of the challenges faced while studying cancer and finding the therapies? Is it that each cancer has its own signatures, or is it that inter-individual variation that every individual has a lot of variation in their own types of cancers? Is it that there are way too many drugs that too many new drugs to treat cancers, or is it all of the above? Yeah. So, is it that there is a lot of variation? Are there too many drugs to treat cancers? Is it A and B, or is it all of the above? So, the correct answer to this question is actually that there is too much inter-individual variation, and it has its own signatures. See that there are too many new drugs is actually not correct. Mm, they actually need to use the pre-existing drugs in new combinations. They cannot really make a lot, like there's not a lot of new drugs being produced in cancer, but they're coming up with a lot of new combinations of these pre-existing drugs in order to treat a cancer. So the correct answer is both options A and B. So that was all for today. I hope this lecture was helpful, in which we discussed what happened during this week in I Think Biology and these some questions. Thank you.